We have a long tradition of doing this. Maybe it helps. So, what I will do here is I will tell the story a bit in the way in a chronological order. So, in the beginning, when we started the research in, I think it was November 2006, we were really puzzled by this experiment of Professor Vietma, and in which you have here the sodium chlorate crystals in contact with the saturated solution, and after applying grinding, the evolution of the single Garl solid phase is found. And actually, to be quite honest, we didn't believe it. We couldn't figure out how it worked. Furthermore, at that time, there was also this paper of Professor Rebo, who said that this would not only work for a system like the agar sodium chlorate, which you have the agar solution and the separated crystals of right and left-handed material, but that it would also work for a system in which the Garl molecule racemizes in solution, like depicted over here, and crystallizes as separate solid phases of both handlers, a so-called conglomerate. So that's what actually, this was the starting point of our research. But we really didn't understand what was going on in the grinding. So the first thing which helped us is to look at the system without any grinding. So depicted over here is, these are sodium chlorate crystals in contact with a saturated solution. And we can distinguish between the two hands because we used cross polarizers, a little bit shifted. And then you see the light crystals over here. These are of one handness, so this one here, these. The darker ones are of the opposite handedness, these and here. And when we wait a little for a while, we see this process occurring. The small ones, like this one here, start to dissolve, also here, for instance, and the larger one starts to grow at the cost of these smaller ones. This process is called Oswald brightening, because the system tends to minimize its free energy, and it can win energy by decreasing its volume to surface ratio. And when we wait a little bit longer, we will see this. All the crystals start to dissolve, and you end up with one single crystal. And this is either left-handed or right-handed. So what you see here is the evolution of a single Garl solid phase without doing anything, just choosing the right system. And of course the outcome of the handedness is completely arbitrary. It's just the largest crystal which will survive. It only takes a very long time. So when we knew this, we start to think, but what could go on when we start grinding? Would that help? That would be strange, because then you still have the process of Oswald brightening, like depicted over here. So these small crystals dissolve, and they nurture the larger crystal independent of their handedness, resulting in larger crystals. And at the same time, you have the process that a large crystal, or a crystal at all, is broken into tiny pieces which have the same volume as the crystal before it is broken. So we were questioning ourselves, when you perform the continuous attrition, will this still give a single Garl solid state? And we start to simulate that using a Monte Carlo modulation technique. And it's called Monte Carlo because it depends on chances. So it's like you're throwing dice every time, and when a process occurs a lot of times, you give it a high probability to occur in time. So this brings me to the outline of my talk. First of all, I will introduce to you the model. Then we will go on to Garl amino acid derivative, like already shown by Professor Vietma. And then we will look back at the model, and we see that it's probably a little bit too simple, and we incorporate some new things, and then we will look back at the experimental results and see if it fits. So first of all, the model. Okay, here's a sort of flow diagram of the simulations which we perform. So at the start, we have solid crystals of both handedness, so left and right handed crystals, We can choose to vary the sizes, but it really doesn't matter. And the model then chooses a crystal randomly. After this, it questions itself, are we going to do an Ostwald ripening step, or are we going to break the crystal in random sizes? When we break the crystal, it's broken into two smaller crystals of random size. However, the volume of the crystals is kept the same, so we don't lose material also. And then we check if the enantiomeric excess is already 100% in the solid phase or not. If not, then we repeat the process. If we are going to perform an Ostwald ripening step, we choose another crystal. And then we compare, then we ask ourselves the question, if we are going to do Ostwald ripening, so is material moved from the small crystal to the larger one, because this is the overall process of Ostwald ripening. We generate a chance for this, depending on the size of the two crystals. And when this occurs and the chance is okay, then we effectuate Ostwald ripening and the process continues. Otherwise nothing happens, and we also continue the process. One of the things which is not incorporated in this model is primary nucleation. I'll come back to this later. So this is just to get some math for people who are not so experimental. What we do is we generate the chance for Ostwald ripening depending on the sizes of the two crystals. So when you look at one crystal, you can see that the free energy for this crystal can be described by an internal energy term, a bulk term, and also a surface term, which is this one here. And as the surface, we assume that these are spheres, like drawn very schematically over here, I realized. And what we do then is that we, when we have two crystals, we compare the free energies of these two crystals, and we generate a sort of driving force for this, to move a growth unit from the smallest crystal to the larger one. And this is simply the difference between those two free energies. And what you see here is a lot of numbers, but these are all constants. It's, for instance, the surface free energy and the volume of the unit cell. It only depends on this. Here we see the number of growth units inside one crystal, and here of the other one, and the difference between those two generates the chance for Ostwald ripening. So if there's a large difference between a small crystal and the larger one, then this process will occur often. That's what it says. So primary nucleation is not incorporated in the model. And we didn't do that because we are grinding in a almost saturated solution, and for nucleation you often need a supersaturated solution. For instance, that's why you make a scratch in the glass when you want to have something crystallized. It's already supersaturated, but it needs a place to crystallize. And for instance, the model, the compound we used, you can undercool it quite a lot before it starts to crystallize. Furthermore, we didn't include it because if we would include it, then these nuclei would be very small and they would immediately dissolve again because of the process of Ostwald ripening. They are very small and there are larger crystals around them, so they would immediately dissolve. Okay, we also have some other parameters which we want to study. For instance, the attrition intensity. So that's, for instance, the amount of glass beads which you add to the flask, or the stirring rate. We treat that as a parameter. So that's approximately the chance between Ostwald ripening and attrition in time. And we also have some non-essential assumptions in this model. For instance, that attrition is only affected down to a minimal crystal size, so very small clusters are not broken in our simulations. This is, however, not essential. It also works when you do incorporate it. And furthermore, in the attrition process we produce two crystals of random size. So you could also break them exactly into equal sized parts. It's not a problem. And this is how the simulation looks like. What you see here on this axis is a crystal size distribution. On this side here, from 0 to 300, are the sizes of the D crystals, and on this side the sizes of the L crystals. Initially we start with two equal-sized crystal size distributions, approximately around 150 growth units. That's the beginning of the simulation. And then we start the simulation, and after a few cycles you see that all the crystals are ground to smaller particles, like depicted over here. And then we continue the process of Ostwald ripening and attrition, and what we see in time, you can see it here on the top view, is that material in this case is moved all to the L enantiomer, and all the D material is completely gone after a while. So it's converted in the L material. And of course we performed many of these simulations, and when you start with equal molar distributions, the outcome of D or L is completely random, so 50-50, that you end up in a D or a L single barrel state. And it always works, so no experiments failed or so. Okay, this convinced us actually that this could work. So it was, I think, in the beginning of December 2006, we started with our model system, because what we wanted to do is we wanted to perform this reaction, so that we had a compound which could racemize in solution, and also which could crystallize as separate phases of the two enantiomers, so conglomerate behavior. This, however, is quite rare. It doesn't happen so often. So we had to design a molecule for it. What you see here is an amino acid derivative of phenylglycine. So here, instead of the acid, there is an amide, and on this side this is a Schiff base called, and it makes it more easy to racemize the compound, because when you look at it, this is the chiral center, and this ring is sticking out of the wall, and there is a proton which is going back to it, and this proton can be extracted by using a base, for instance DBU, and this removes the proton here, and this structure is then stabilized by the Schiff base, and the proton can enter the molecule again either from the top side or the back side. And when it enters from the other side, you will get the mirror image of this molecule, and this is how the racemization works. The other problem is that we have to form separate solid phases of both the S and the R compound, and we could do this by engineering this part of the molecule. So what we did is we built a library of compounds with many substituents hereon, and we looked to find conglomerate behavior. And this is the molecule which does it. Okay. This is how the experiment looks like. So we start with racemic material, partially dissolved, so we have racemic D and L crystals in contact with a saturated solution. We have here the glass beads and a steering bar, and in the beginning we just stir for 24 hours to make sure that we have equilibrium. After that we add the DBU, so that's the base which makes sure that the racemization is going on, and we simply apply the stirring. And in time we take samples, we filter it, and the solids we analyze them using chiral HPLC. So chiral HPLC can distinguish between the two molecules. That's the way we analyze the material. Okay, so what you see here is a graph of the enantiomeric excess in the solids in time. So in the beginning the material is racemic. However, after a few days you will see a slight increase in the enantiomeric excess. And when we wait long enough, you enter a completely enantiomer pure state, like depicted over here. However, in the beginning it took quite a while, almost a month or even longer. And these experiments were performed with several batches of starting material, because we really wanted to make sure that people would believe our experiment. And furthermore, we performed them in 4 different laboratories, in Nijmegen, Geleen, Groningen, and also in London. And what you can see here is that all the experiments act quite the same. The behavior is quite similar in all the experiments. And it's very reproducible. Myself, I performed more than 100 experiments and they all act like this. What furthermore puzzled us in the beginning was that all the experiments went to the hour enantiomer. And that was really strange, because we started with racemic material and you would expect the outcome to be 50-50 towards the R or the S enantiomer. And that was a big problem for us in the beginning to what was going on. So we were thinking of a possible source of contamination which was causing all our experiments to go to one-handedness. To check this, we used additives. So in this case, what an additive can do is, like depicted here, I think we have already made one such a schematic drawing. What you see here is is a crystal of one-handedness, and this is of the mirror image. And an additive, which is enantiomerically pure, can be built in one of the two crystals. So this additive fits in this molecule, in this crystal. However, it doesn't fit in this one. So in this way you can stereo-selectively influence the crystal growth or dissolution. And as an additive we chose phenylglycine, enantiomerically pure, hoping that it would stereo-selectively hamper the growth or dissolution of one of the two, and thereby controlling the outcome. So here is the result. Let's start at this part of the graph. So again, here you see the enantiomeric excess in time in the solid phase. What you see here is when we add different amounts of concentrations of S-phenylglycine. So it's the additive I just showed you. And when you add a bit of it, so only 0.1%, the reaction just goes to the R. This is still the enantiomer we also got from racemic material. However, when you add higher concentrations of this, the process is enhanced. And the time is reduced up to, approximately 6 days, which makes it even more relevant when you want to talk about the origin of life. Helps to convince people. And you can do it within a week. But then, this is the natural handless of amino acids, the S one. Now we added the artificial one, which is the R enantiomer. This one does not occur in nature. And what you see now is that you end up with the S-garl solid state of the compound. So we can control the outcome to both sides using very small amounts of R-phenylglycine or S-phenylglycine. And at this point, I think it's very important to realize that when you're performing an experiment, you're performing this experiment in a garl world. I mean, somebody did it already for us, and this influences all our experiments. And what we see here is that even minute amounts of S-phenylglycine or R-phenylglycine already overrule the contamination we are dealing with, and can control the outcome. So it's very likely that our experiments are contaminated with natural amino acids, proteins, or anything else which is garl. So it works. So what actually happens is the growth or dissolution of is hampered stereo-selectively, so not for this one, and this for this one. And what you see therefore is that the growth of the S enantiomer in this case is hampered, and thereby you end up with the R end state. And this can be compared with the rule of reversal, which was already depicted out a long time ago by the group of the Weizmann Institute. And they have the same model for this kind of behavior. But why are the concentrations so low? Well, you don't need to block the whole crystal to grow, you only need to block the active growth sites. So what you see here is a drawing of a crystal surface, and what you see here is growth steps. So normally when, especially under near equilibrium conditions, material comes on the surface and is incorporated at special places at the crystal. And especially at the kink sites, which is here. It's a sort of corner, and there the molecule can be built in. And only these kink sites need to be blocked by the additive. to have a good effect. So therefore the concentrations can be quite low. Although it is of course true that by applying the continuous grinding of the crystals they will be a little bit more rough than normal. So then we did another experiment. We want to show what would happen when you start, instead of racemic material, with a slight imbalance in the enantiomeric excess in the solid phase at the beginning of the experiment. So here we start with 3, 5 and 10% initial enantiomeric excess in the beginning, and you see that the higher the enantiomeric excess in the beginning, the faster the experiment ends up in a single Garl solid phase. And we can do it in both directions. So even a small enantiomer imbalance already directs the outcome of the experiment. Okay, this is always should happen. Well, I wanted to show here some microscopy pictures of the crystals. But yeah, this one is with cross-polarized. But what you can see here is that it's difficult here. Maybe I have a better picture, but this is after the grinding, and what you see is that the crystals are still quite. The morphology is quite good. They really have distinguished crystal surfaces, and furthermore, you can check using the polarizers on the microscope. You can check if they are still made of one single crystal, and they do. You can see it here. Yeah, I have the other picture here. Then you can see that this one here is lightened up completely. When you turn the polarizer for 45 degrees, the crystal is completely dark. And that means that the axis is in one direction.
How long after grinding?
This is about, I think, 10 days.
After grinding.
That means we ground the sample for 10 days,
Yeah.
then we filtered it off, and then we measured it.
So you filter it out how quickly after you stopped grinding?
Immediately. I think that is 10 seconds or so. I need to get a pipette there and take a sample, and then so that's the fastest you can do. Yeah, of course you want to look in situ or so to see it. That would be even better. However, at the moment that you start to filter it, the liquid is immediately withdrawn from the sample. So okay. What I now want to do is I want to look at the model and verify it with the experimental results. And one of the first problems we had is that when you look at the model here, you see that the evolution in time of the enantiomeric excess is quite stochastic. For instance, here the system is almost finished, but then it moves on again a little bit towards the D enantiomer before it ends in the L1. And when you look at the experiment, you see that it's very deterministic. So once the symmetry is broken in one direction, it will be amplified in that direction. So actually, the model was a bit too simple, which we used in the beginning. So what we actually did in the model is that the transport of material from a small crystal to a larger one had the same rate as the transport of growth units from a small crystal to one of the opposite handedness. So actually there was no distinction between the solids. And this maybe was not very realistic, because when you look under experimental conditions, for instance the racemization will not be instantaneous, and also Garl clusters might play a role. And therefore we distinguish between a sort of homogarl transport of material and heterogarl transport of material. So in a homogarl transport, material is going from one crystal to one of the same handlers, which can happen without any difficulty. On the other hand, when material is moved from one crystal to one of the opposite handlers, which we call a heterogarl step, then you first have to, in case of our racemization reaction, the molecule has to be racemized. And in the case of Garl clusters, a Garl cluster has a certain handedness. It can be built in a crystal of the same handedness. However, it cannot be built in a crystal of the opposite handedness. Therefore, the the cluster first needs to be dissolved, and then it can recrystallize on whatever. So, and we have a certain parameter for that. We call it the racemization efficiency, because that is the rate at which this racemization for the material can be calculated. Then in this situation it's instantaneous, so it's 1, and when the racemization is slow, so this step, then it's smaller than 1. So we only had to change the model a bit, because this part is exactly the same as in the beginning. However, when we choose the second crystal, we now look if it has the same handedness as the first one, or if it has the opposite handedness. If it has the opposite handedness, then we make it a little bit more difficult to transport material from the small crystal to the larger one. Because, yeah, you can have a gargle cluster, or you can have problems with the racemization hampers the growth. So therefore, we adjust the chance for Ostwald ripening a bit in this case, and then we continue the model. And this is the result. So this is the picture which I showed you in the beginning, and this is when you apply this racemization efficiency. Again, we start with equal amounts of solid material. What you can see now is that once the symmetry is broken, the evolution of the enantiomeric excess proceeds smoothly towards the handedness, towards the enantiomer of towards the major enantiomer in the solid phase. So what you see here actually is that for the values of chi which are smaller than 1, you see that the behavior is deterministic instead of stochastic. So let's take a look at this a little bit more in detail. Let's first focus on this line here. What you see here is the racemization efficiency as a function of the deracemization time. So that's the time for which it takes to start from the initial state to go towards the single gargle solid phase. And when you have instantaneous racemization, so chi is 1, you see that it takes quite some time. And this is because the system is spending a lot of time because it's moving stochastic. However, when chi is becoming smaller, so the difference between heterogargle and homogargle exchange of material is becoming larger, then you will see that the deracemization time drops dramatically. And after a while you see that once the racemization, for instance, is very slow, you will see again an increase in the deracemization time, because this is now becoming the rate determining step. Furthermore, what we also wanted to verify is the deterministic behavior. So what we did is we started in the beginning not with 0 enantiomeric excess in the solid phase, but for instance with 5% excess in one enantiomer. And when you have instantaneous racemization, then the chance to end up in the solid phase, in the single gargle solid phase, is 5% more than for the opposite enantiomer. So you always end up in the single gargle solid phase. However, 5% more times you end up in the D enantiomer, so the initial excess. And this is more. This is amplified more when you move to slower deracemization efficiencies. So when you racemize in this regime, so quite slowly, you will see that almost all experiments end up in the enantiomer which is initially more abundant. Furthermore, we also looked at, already mentioned that we can look at the influence, for instance, of attrition. So how much attrition do we apply to the crystals? And that's written here. So at this part of the graph, we do not apply attrition, and a lot of Ostwald writing is occurring. And as I already told you in the beginning, this process is slow. So the deracemization time, it takes a long time. Notice the logarithmic scaling here. However, when you apply more and more attrition, the deracemization time is decreasing, like here. And this is because you form small clusters which dissolve more easily, and the whole process is faster. However, at a certain point in time, it doesn't work anymore, because at this situation here, the de-racemization time is increasing again, and this is because we only have clusters of the minimal size. So we had a sort of minimal size for attrition, and all the clusters are now so small that there is no driving force between the clusters anymore. Okay, so this explains why you have to grind so much. Okay. Furthermore, we can also look at the evolution of the enantiomeric excess in time. So when we start with a small imbalance, for instance 2.5% enantiomeric excess in time, then in the beginning you still see some stochastic fluctuations in the computer model. However, after a while, the system smoothly runs away towards the single Gabel end state. And the more initial excess you apply to the system, the faster this works. We were able to calculate a sort of first order kinetic description of this, in which you can see that the enantiomeric excess at a certain point in time depends on the enantiomeric excess in the beginning and an exponent of a rate constant time at the time. So when this would be true, then therefore we plotted the logarithm of the enantiomeric excess in time. And what you can see is nice linear behavior. Here you can see the stochastic behavior which was in the beginning of the small enantiomeric excess. And then we want to verify, of course, if we could also observe this kinetic description in the experiments. So here again the graph of the small imbalances in the beginning. And when we plot here now the logarithm of the enantiomeric excess in time, again we see the nice linear behavior of the graphs for different enantiomeric excesses. So what we see here is that the kinetic behavior is the same in the experiments as in the computer model. Okay, what we can also look at is the Racemization parameter which we introduced in the model. And this one is actually in the rate equation here, in the rate constant. So let's take a look again at this graph which I showed you. And actually, when we are doing the reactions, we always are in this part of the graph, because it's always deterministic. So the small imbalances are amplified very well. And what we now did is that we wanted to fit a parameter to this. So what we did is, instead of the Racemization time, we plotted the inversely depending parameter of this. And what you see here is nice linear behavior. So you can write that the de-Racemization time is inversely proportional with the Racemization efficiency. And from this equation you can write that when you take the enantiomeric excess at time t as the enantiomerically pure state, you can write this equation, so that you see that the rate of this de-Racemization process is linearly dependent on the Racemization efficiency of the process. So now we want to verify if we could measure this in the experiments. So the first thing we have to check is, so what you can do then, for instance, is add different amounts of base to this system, because the base does the Racemization. The first thing you have to check is how is the concentration of dBu depending on the amount of in the solution. So how is this kinetically described? So what you see here is only the solution. This is a saturated solution without any salt. And what we do is we start with an enantiomerically pure material, and we add different concentrations of base. So what you then see is the Racemization reaction, and you end up with zero enantiomeric excess. And what you see is, for different amounts of bases, the Racemization reaction is faster for large amounts of base. What we now can do is we can calculate the rate from these from this evolution of the Racemization. And what you see then is that the rate of this process linear depends on the concentration of dBu. Okay, so what we did then is we performed de-racemization reactions. So this is with the solid and the solution which is racemizing for different amounts of base, like depicted over here. And what you now can see is that the de-racemization increases for larger amounts of base. So the faster you perform the racemization reaction, the faster this process will occur. What we now can do is we add this kinetic description, so we can fit this description and measure the rate constant for this de-racemization process. And this is plotted over here. So for the concentration of dBu, we measure the rate constants, and as you can see, it's quite smoothly a linear behavior. Not exactly, actually it goes a little bit like this, but okay, this again is quite well described by the computer model. So what we actually can conclude from this is that the racemization is quite slow in comparison with the exchange of material between the solids. So this de-racemization parameter, it can be really observed. Finally, I would like to say something about the driving force of this whole process. And when you take a close look here at, this is again the top view of one of the simulation data I showed you, and what you observe here is a sort of shift of, in the beginning you see the amounts of, the crystals have all the same size. However, at the end of the process you see a sort of shift towards larger crystals. So the crystals become larger. That means oswald writing becomes more efficient. And what is depicted over here is the total amount of crystal surface of both the D and the L enantiomer in time. So what we first of all see is this simulation is moving towards the L enantiomer. So what you see is that the total amount of crystal surface in the L is increasing. And this is because although the crystals have almost the same size, you see that, yeah, there is more material, so the total surface will increase. Similarly, you will see that the amount of D enantiomer decreases in time, and therefore also the surface of this D enantiomer. However, when you summate these two things, you will see that the total amount of surface is decreasing in time. So that means because the total volume of the solids remains the same, it means that the particles are larger. And this is because oswald writing is becoming more efficient for this increasing enantiomeric excess. Because in case of clusters, they will find more easily a crystal of the same handedness, and therefore they will grow larger. So I would like to end up with some conclusions and implications. So what I showed you here is that using oswald writing, you always end up in a single Garrel solid phase, and this can be enhanced by applying attrition. Furthermore, what we see is that this computer model quite resembles the experimental results very well, and I showed to you the first practical proof for Garrel molecules of this. Furthermore, which is of interest, is that we can control the outcome of the de-racemization process using, for instance, enantiomerically pure additives or very small initial imbalances in the population between the D and the L enantiomer. And well, one of the implications is, of course, as already pointed out by Professor Vietma, of course, is that this can have implications for the broken symmetry in the prebiotic world, for instance, the amino acids and also sugars. Furthermore, it's also very interesting as a new way to prepare enantiomerically pure compounds, because, well, we saw some examples in the presentation of Professor Vietma. About 80% of the molecules which are now used in pharmaceutical products are Garrel, so they need to be prepared in a Garrel way, and this is a very attractive method to do it. One of the things, for instance, is that you have 100% yield, so what you put in the de-racemization process comes out. You don't lose material. Therefore, it's very interesting for pharmaceutical applications. I would like to end this presentation by thanking all the people who worked in this work, in this research. First of all, DSM is this is a company, very big, and also the company Simcom in Groningen, the University of Nijmegen, and also Imperial College, the group of Donna Blackmont. Everybody contributed to this work. And I would like to thank you for your attention. Thank you very much. This is just a photo just after we discovered it. It's in January 2007. Yeah, as you can see, we are all very happy. Yeah.
Questions? How many of you were left-handed?
I don't know actually. Yeah. For instance, one of the things we did check is if the steering, if you apply it to the left or to the right, if that would make a difference. On a certain moment, we both realized that somebody could ask that question, and so I wanted to go to the lab to do it. But it turned out that at DSM they were all turning to the left side, and I was turning to the right side, depending on the kind of steering which you used.
So you did the experiment already?
Yeah.
Henry?
Since you are speaking of applications,
Yes.
what is patentable in this type of
Yeah, it's very difficult actually. One of the problems actually is to check if somebody is using your method, because it's a very clean method. You don't need any Garl compounds in the beginning. For instance, when you perform an asymmetric synthesis or so, you always add Garl catalyst or so, which leave traces in your product, and which you can detect. However, in this case, you don't have that. So the difference between, for instance, performing a sort of experiment where you start with a clear solution and then you crystallize it out, or applying this technique, you cannot see the difference in the product. So therefore, it's very difficult to have a patent on it and check if somebody is doing this. That's one of the great problems, actually. Yeah, that's very difficult to verify if somebody is using your...
Have you patented?
No, it's not patented. No. So everybody can use it.
Did you share samples among the 4 labs that all got R? You said 4 different labs did the experiment, all got R. Were samples shared? Or did you
but only after the first results came in, because we were quite afraid of contaminating each other.
Yeah.
So what we also saw is that we prepared starting material, for instance, in different labs, and performed the experiments with those starting materials.
But still, it was all. So that's a little surprising that it's the same impurity, analogous impurity.
Yeah, well,
4 labs.
But I think it's not so strange when you realize that the whole world is made of one enantiomer.
Yeah,
But
what makes me dirty might be different from what makes you dirty.
Exactly, of course. We put a lot of energy, actually, or effort in it to find what could be the thing which causes the imbalance, and we never could find it.
Yeah, but you were able to show that with a very small amount of artificial disturbance you can turn it the other way
You
around.
actually got to see pharmaceutical.
Sorry?
When the bottom line, novel methods, you
Yes.
can't actually start from the...
Yeah, of course. When you
have to see it.
that
Therefore
is.
you may be able to detect it.
How do you mean that?
You said you couldn't detect it because it started from purely racemic.
Yeah.
But actually, if you did that, you'd get your problem.
So yeah,
Right.
I think
I'm not sure if
go back a few slides where you had the bifurcation to up and right and left. Yes, exactly.
Oh, this one. Yeah.
So all you're saying is you should be homing in very close to
You've
0
got
to find exactly
the
what.
original problem of
Yeah.
impurity.
Okay. Yeah. Well, what we did is from the other data. Let me show it. For instance, what you can yeah, it's a little bit slower than I am. what we did is, for instance, we fitted we calculated the rate of these different lines, and from this you can extrapolate the initial bias which is needed, and it's about 10 ppm. So that cannot be detected by any way.
I mean, the rate is the same, the offset is different.
But
Yeah.
what I'm saying is a chemical company doesn't want that.
Oh,
Chemical
yeah,
company
but
wants
when you,
certainty.
yeah, okay, but
And
then,
so they have to.
well, when we are talking about that, I would perform the experiment completely different, because I can show it to you. We actually tried this on large scale, so just to make sure if it remained the same, and it was. But when you are a chemical company, yeah, we are here talking about the origin of life, but okay, money also works. Then you don't start Racemic. What you do is you start with an initial imbalance already, because it's an exponential behavior, so it's faster once you have an initial imbalance. Yeah, exactly, and half of it or so you throw back in the reactor and you do it again. That's the way you want to do it, and then you always end up with the correct handleness.
There's another point I want to point here, that most of the racemic amino acids which are generated in companies are made by racemization of the L amino acid to the amino acid. And
Yes.
presently Schurig and I thought actually the problem with tyrosine, Schurig in Tübingen actually investigated the composition of various racemic amino acids and always find a small excess of the L amino acid in the water.
Yeah, and that's because they are made in this way. I think so, yeah.
Because you cannot reach the racemization up to the end. Now it's always the end with an excess of the elements.
Okay.
The other question, let me just ask one thing, as you have this picture.
Yeah.
This offset here, 4.06,
Yeah.
do you think that could be due to osmo reddening then?
No, I think actually that this line should be a little bit more like this.
Yeah.
And that for these very high amounts of DBU, it doesn't act linear anymore. Yeah, it's quite a lot. What's that?
Yes, well, that is the principal point is about with the Racemic picture. I was wondering that in the publication in Czech is not spoken about.
Yeah, it's only slightly incorporated. Yeah.
And I was expecting that this is the answer, that you obtain always the same.
Yeah, I can go a little bit.
The influence of cryptochiral combinations in this symmetry breaking process is very, very strong.
Yeah, it's very likely.
We have sent some months ago communication, chemical communication, it was not accepted because we could not identify the contaminant.
Yeah.
And I could present something later on this one and I thought you will. But I was supposing that your result was just with the Racemic picture and always the same sign.
Yeah.
Only to say that in the Czech society, you don't speak about the Racemic.
No, yeah, well, we were also afraid of that referees or so.
Well,
would
but
make
I was wondering that the referee is not asking about this important point because
Well, it was included in the checks, but only in a footnote.
Homochirality without to speak about it.
Yeah, it was included only in the footnote. So, but I can say another thing actually about the bias, because we now were talking about that it was coming from probably a contamination. Alternatively, it could also come, of course, that your material is not completely racemic. Very bad. So, what also could happen is that it's not completely racemic, and then you.
But for example, when you start with with this other compound, you don't have the same. You don't start at the same concentrations in one direction as the other one.
No. No.
And then it agrees with the sense that you obtain with the Racemic.
Yeah. But we can also check this actually.
It was also not indicated.
Yeah, I think what we described in the experimental section is that we can do several things to check that this is not going on, so that we don't have an initial imbalance in the solid phase. One of the things which should be noticed is that our molecule, the starting material for it, is a very stable racemic compound. A racemic compound, so that means that you have both enantiomers in the unit cell. So the starting material is therefore completely racemic. It's as racemic as it can get, of course. I mean, the chance for having a completely racemic sample is about 50%. You need an even number of molecules. Furthermore, what we can also do is, because this is a conglomerate, you can use the trick which Professor Kjepma already pointed out, is dissolving a lot of material. Because what will happen is that the solution concentration will remain racemic, and thereby, for a conglomerate case, the solid material will enrich in the enantiomer, which is more present in the beginning, and then we could detect it, but we also didn't find that. So I think that the material is very racemic, actually. Yeah.
Yes, please.
As for a question, with your very low reaction times, do you observe any analysis of the
Yeah, that can be a problem, actually. For protic solvents, so initially we did everything wrong, actually. We started with a bad solvent, and after that we used methanol. This is how we make it. But, yeah, this can occur. In the solution phase, you can have hydrolysis. So that means that you get a proton from the solution, which is donated here. This one is from the solution, and you get this ring system. And this ring system, you can have two diastereomeres, and also the mirror images, because from the other enantiomer. This is formed in the solution only, of course, for protic solvents like methanol, and it's completely racemic. But yeah, it can be a problem. Therefore, we switch to other solvents. So, for instance, dry acetonitrile, which are the results I showed you, doesn't have that problem. Yeah.
What's known experimentally about how grinding works? I don't know anything.
Yeah, it's horrible.
For example, you stop grinding at a given point. I assume that in your model, and I assume that the reason for that is that so you don't get too many particles to keep track of. Is that right? Or why do you stop grinding?
Oh, when?
In your computer model,
yes,
you have a minimal size.
Oh, so we just do that. You don't need to do it. It also works when you just grind everything up to up to the smallest level of growth units which you have.
Why did you decide to do that?
Yeah, because we thought maybe it's experimentally more. Well, what we think is that you don't grind very, very, very small crystals. I think maybe you do, but okay. Yeah, I also don't know.
You know, there must be people who make flour in Holland and that somebody must have studied grinding. I don't know anything about it.
Yeah, well, normally you end up with a sort of crystal size distribution which I showed you. So I think you always end up with the large particles and also the small ones. And I think, yeah.
And then, in the same connection, if I understand it, when you decide to break a crystal in grinding, you choose arbitrarily the size.
Yeah, we had a long discussion in our department on that.
So the problem, it seems to me, with that is that how do the crystal, it seems to me maybe more likely that grinding takes small bits off a crystal. It never breaks it in half or even in thirds or something like that. Or maybe it does, I don't know.
Yeah.
But these are differences. And do you have a basis for thinking that you're, not that necessarily it depends on this, but do you have a basis for deciding that when you grind you will break a crystal in half at an arbitrarily chosen boundary?
Yeah, the basis for this is that it is simple. And I completely agree with you that you can make this more difficult and more like in real experiments.
Okay. Always to take off particles of a given size, for example.
Yeah, that's also possible.
And I don't know how grinding works, which is more realistic. I think the result is qualitatively similar.
I can recall that in old days, when it was very difficult to make X-ray structure analysis of crystals, people were trying to correct for absorption of the X-rays.
Yeah.
And I remember that the way to find that was to try to grind the crystals and to get balls. Actually, you're not breaking single crystals, but you're getting actually round balls. And what you have kind of, you are somehow peeling some surfaces of the crystal family that have some, but you're not cleaving it into different, of course, depending on the way and the problem. But
I think so.
Something else that makes me think that is that you say that these little crystals have nice facets, you can identify facets. That sounds to me like you're not breaking them, but just taking a little bit off, but it's getting put back on all the time, so it keeps sort of the same shape.
Yeah, that could very well be possible. On the other hand, I think it also much depends on the equilibrium morphology of the crystal. For instance, our crystals are a little bit needle-like, and sodium chlorate is cubic, for instance. The breaking must be different. And still, we observe the same behavior. So I think, of course it is important. And I think for a detailed study, this should be accounted. Yeah.
Have you looked for literature?
Yeah, we did. And I think it is so specific for each compound that it makes it very difficult. Yeah.
Just because you check the effect of addition of amino acid for your page. Did you check when it's starting amine? Because that's where
also. Yeah.
It's a reversible process, maybe information.
Yeah.
Since acetylation even takes up lacks water salts, probably formal.
Yeah, we checked that and it didn't do anything. Actually, I also have a sheet incorporated about how we think approximately how it works. Because it's a little bit slow, sorry. Yeah. Okay. The crystal is built out of molecules which are very neatly stuck after each other. And what you can see in the crystal, this is why it forms a crystal, is that it can have bonds between each other and thereby wins energy. And our molecule is an amide. And this is published already a long time ago. In a study for additives, what you can see here is the amide bond, in which the nitrogen here shares a hydrogen atom between a molecule in this chain and also one in this chain. And what they then did is they had a little bit of benzoic acid, which was added as an additive, and what you can see there is that in this case it is built in very nicely because it's almost the same structure. But what you can see is that it can only donate, because it's an acid, one proton to this chain and not anymore anyone to this chain. And therefore the growth in this direction is hampered. And actually I think that our impurity, so the acid which I add, acts a little bit like this. Of course, we need to do more study on this, but this was actually why I chose this MeOH acid for it, and not an amide, which would be more logical in the beginning. Yeah.
August, could you come back to the previous slide? He was on the board.
This one?
No, the just before. With the mechanism of the racemization.
Oh, that was really. I don't know how this worked. That I don't have to skip all the.
Yes.
Oh, this one.
This one.
Yeah, but this is the cyclization, huh?
Yeah.
Okay.
That's okay. Just in the molecule which was on the left. If you, instead of amine, if you use ester,
that would also.
It works also very well.
Yeah, of course, because you only don't want the acid, because then you start
and the extra proton on the chiral center is removed very fast too. And if you use not prebiotically speaking, if you use an enzyme in this situation, you can move very fast in amino acid, in only one amino acid, L amino acid, if you want.
Yes.
With an enzyme which links only the esters, the L ester.
Yeah, you mean a sort of dynamic kinetic resolution?
Yes, exactly.
Yeah.
The dynamic resolution.
Yeah.
And it works very well.
I think so, yeah. The fact...
I'm sure.
Yeah, okay.
We do.
I think in DSM they also use this as a process to make it using an enzyme. They also do these DKR reactions. Actually, we didn't choose for the ester because then in the crystal structure you will have less strong bonds, we guessed, and therefore we chose for the amide and not for the ester. Yeah, but that was the only consideration. Sorry?
Should work also with the ester.
Yeah, I saw some results now.
Now I can make a proposal. I propose you to apply such a reaction to a French population. Instead of deracimizing the French population only in one side or in another one. So there will be no fight between the boss.
Oh, yeah.
Population.
Oh, I was thinking more about performing the same experiments on the other side of on the south side of the Earth, where the weather is good.
Thanks to apply.
Yep, thanks very much. Lunch, it costs 56 kroner for the base lunch. Any extra drink costs more, and also bread costs more. So 56 is the minimum price. Thank you. So we resume and reconvene at 2 o'clock.
Did they ever give you the presentations?